talks on toxoplasma in the past couple of days. So I'm just going to say something about the developmental aspects of toxoplasma here. So this picture, it shows how toxoplasma goes through the two hosts, the primary host and the secondary host. The primary hosts are invariably feline species, and it has to be a naive feline species to support sexual development in the gut. And once the parasites develop, they come out as oocysts, stay in the environment till they are picked up by secondary hosts. And in the secondary host, they have the choice of growing as tachyzoids, which are virulent, can kill the animal if given a chance, or they can actually differentiate into this latent bradyzoid cysts, which can stay in different locations in the animal system. So this is sort of the life cycle, and there's a lot of interesting things here. First of all, this parasite, unlike many other parasites, have learned how to break the species barrier, which means that this one species can infect multiple hosts. In fact, any warm-blooded animal is a host for this parasite. So it has learned to live with so many different hosts, so many animal hosts, and it has adjusted to the immune response from different hosts, and it has to also develop in this host. So I'm not going to talk about any host parasite interaction today. I'm just only going to talk about the parasite aspect, how it develops through its life cycle stages. So, like I said, there is this two asexual in asexual growth. It has these two stages. These tachyzoids, they proliferate very rapidly. They invade a cell, and then post invasion, they rapidly replicate, and then they make hundreds of parasites. They lyse the cell, and this cycle continues. They can switch to this latent stage under some kind of a stress. The stress can be an immune stress as well, and then when they invade, they just stay. They don't lyse and come out, and they can stay for many years like this. And actually, this whole process is reversible. So given a chance, these bradyzoids can actually come back as tachyzoids and cause severe disease. So this is a problem in immunocompromised humans. Actually, toxoplasma is not a killer disease that way like malaria, but in certain conditions when the humans become susceptible, this can happen, or for that matter, animals as well. Now, if you take the sexual stages, I just want to walk you through this slide here. So this is the gut epithelium of the cat, and once only the bradyzoid stage of the parasite can go through the sexual cycle. Tachyzoids cannot, so it's like a one-way traffic here. So once the bradyzoids get in, they undergo this merogony, and then there is all this enteroepithelial stages where the gametes are formed, the male and female gametes, and then the zygote, and then oocyst is formed. And the oocyst comes out in the environment, they sporulate, and then the sporozoids stay ready to be taken up. The interesting thing here, we'll come back to this in the end of the talk. You just remember this, because these stages have to stay in the environment, they have a very thick wall. This is like a cyst wall, and it is made up of complex metabolites. The bradyzoid also has a very thick wall, maybe because to avoid immune interaction with the host. This is not extracellular or environmental, this is environmental, but still it has a thick cyst wall here. So this is just again to summarize that, we go this whatever you see here, this is reversible, but once you go to bradyzoids and when you go into oocysts, so this is all one-way traffic here, it is irreversible. So, and people have been trying to understand the molecular mechanisms involved in this kind of stage switching and all that, and mostly the problem is, of course, sample availability. So these parasites, these tachyzoids, we can grow in the lab. Bradyzoids, we can induce formation of bradyzoids in the lab, but we can't get mature cysts in the lab. We need animal models, and mice is a good model to get these bradyzoids. But then we have to take it out of the brain, or in case of rats, we can get it from testis as well. But then we really don't have a good system for this oocysts. There were labs which used to work on cats. I think now very few labs, and I think in the US also they have stopped doing cat work. So it's very difficult to get oocysts. So by and large, our understanding has come from RNA studies. So people looked at gene expression to see, in whatever samples they can get. So this is just data from ToxoDB, the genome database. We have lots and lots of RNA-seq data there. So you can do comparative analysis. So we did that. So you can see here that this is the x-axis is all tachyzoids and the y-axis are different stages. And you can see that bradyzoid looks very similar to tachyzoid expression-wise, but there are some small changes, but significant changes. But these enteroepithelial and oocysts look very different. So there's a large amount of change going on in gene expression. And so how are these gene expression governed? So there are lots of many mechanisms proposed. Mainly it is just in time mRNA expression, just like how you see in malaria as it goes through the different stages. And then you have transcriptional control, and these transcription factors called AP2 factors, they play a key role. Okay. So our interest was all metabolism, because we are not a transcription lab or epigenetics lab. So I wanted to see what are the metabolic enzymes, how they behave. So if you take all these genes, so for example in this particular plot, what you are seeing is all the genes that are either upregulated or downregulated in bradyzoids. This is 295, of which there are some significant 31 genes which are enzymatic genes, so enzyme coding genes. So we took all this and we were doing this analysis, and it was interesting to see that in this enteroepithelial there's a lot of enzymes involved in lipid and fatty acid metabolism, which is getting upregulated or modulated. So if you look at this pathway of fatty acid biosynthesis, everyone knows here, I think there is this type 2 in the epicoplast of epicomplexans, but some epicomplexes, including toxoplasma, they have type 1 also, which is like this multimodal fatty acid synthase. In addition, they also have fatty acid elongation happening in the ER, so they can scavenge fatty acids and elongate. Where exactly this fatty acid synthase 1 is located is still like a it's not known, because it's I'll tell you why it's not known in a while. So we were interested in this fatty acid synthase because it came up during sexual stage differentiation, and we were from the genome data we can see that this is a huge mega synthase. It has multiple repeat modules. It's actually very big, 10,000 amino acids, so doing biochemistry will be difficult with such a big protein. And there's some data now from sequence analysis, as well as one paper on functional studies, that suggests that it may actually not be a fatty acid synthase, it might be polyketide synthesis, because polyketates are also required for complex lipid biosynthesis and things like that. So when we looked at the expression of this, so this is from data already available in the genome database. In the tachyzoid stages, there is not too much of expression. So you can see here I put the sense expression from this gene. So this is the entire gene, and then this is the antisense expression. So if you look at the expression, you see that sense is very less or absent in tachyzoids and bradyzoids, but there seems to be some kind of an antisense coming from the 5' end of this gene. And whenever this sense is expressed, this antisense disappears, and then again when it goes to the oocyte stage, the sense is again going down and the antisense is coming up. So it seems like they are contradicting each other. And so this is all data from the database. So what we thought, first we have to establish, sometimes what happens, we grow parasites in the lab for many decades, and we think that whatever is reported happens in our system also, and we spend a lot of time working on our parasites and we realize it behaves differently. So we first wanted to establish that in whatever parasites we are culturing in the lab, we have similar kind of RNA expression. So we did that. So this is RNA-seq, and here you can see very clearly that the antisense is there, but not much of a sense expression. So we characterize the promoter. So what we did was we put this mCherry gene in the first exon in frame, and then we wanted to see why is the sense not expressed. Is it because the promoter is not active, or is some other mechanism? So when we did that, we found that mCherry was expressed, so which means that this promoter is active. And independently, other studies have confirmed, some genome-wide promoter analysis studies are available now in Toxoplasma, which have confirmed that this promoter for this gene is constitutively active. So despite that, the gene antisense is not getting expressed. So we were obviously interested in looking at whether this antisense has any role in suppressing the gene expression. So the first thing we did was we took this antisense sequence, we made sure it doesn't encode any protein. So there are tools now to look at what is the coding potential and things like that. So if you compare it to other very well-known antisense long non-coding RNAs, so these are like, the scores are all very comparable. So this RNA looks like it's a proper non-coding RNA. It doesn't code for any peptide. So then we wanted to see what's the mechanism by which this is expressed. Is there a promoter for this to express? So we cloned out this region and put it put it in front of a GFP cDNA, and then we inserted this cassette in the UPRT locus of the parasite, and then we could get expression. So we have identified the promoter also for this. So there is a promoter driving the expression of this antisense. So then pretty much we thought, how do you do the functional analysis? So the first thought that came to our mind was, let's just knock out this region and then see if we remove the promoter region, whether this antisense will go away, and whether that will then allow sense expression. Of course, if you delete here, you are also deleting the sense coding region, but we are not interested in the protein. We just wanted to see if RNA can come up. We're just studying the expression mechanism here. So we cut out this region with 2 guide RNAs, and we inserted some selection markers here, and we could get the knockouts, and you can see that the RNA disappeared, the antisense disappeared, but we could not see any sense here. So basically, the promoter is active, the antisense is removed, there is no expression of antisense, but we could not get the sense also. So this is where we are with this expression analysis, and we still don't know the mechanism, how this gene is repressed during tachyzoids and bradyzoids, but expressed only during the sexual stage. So we thought that there must be a transcriptional factor, which is, although this is a constitutive promoter, there must be other internal repressors binding somewhere internal in the gene, and then that is how the mechanism is. So we are looking for it, but we still don't have that answer. But in the meanwhile, after knocking out this RNA, we saw something interesting. We saw that the parasites were growing, very slowly, and you can actually see that they are lagging behind in their replication speed also. So their cell cycle is taking longer, and they are lysing out later. And we can see that if you do plaque assays, you see very, very tiny plaques that take a very long time to grow. You really have to look at the plaques using microscope, whereas in wild type you can visually see the plaques. And because these the vacuoles were looking more like bradyzoid cyst forming vacuoles, we were curious, so we did this staining with this reagent called dolichos bifluoros agglutinin. So it basically binds to carbohydrate containing proteins, which are deposited on the surface of the cyst when the bradyzoid cyst is formed, tissue cyst. So surprisingly, even under normal growth conditions, these mutant parasites were depositing this marker, whatever lectin markers were getting, the lectin binding markers, glycoforms. When you give alkaline media conditions, even the wild type will do this kind of deposition, but and the mutant is always doing it. So that was kind of interesting. So we thought some rewiring has happened in the transcriptional control of this marker expression. So this is supposed to happen only during Brady's White formation. So then we took these mutant parasites, we carefully saw from invasion, that is, you allow the parasite to invade a host cell, and day wise we were saying how the marker gets deposited. So this is like uh, it's not the same vacuole, we couldn't follow, but it's just the same culture. Like you can follow when the vacuole is very small, and then by day 2 3 when the parasites are increasing, initially all the glycoforms which are secreted are all internal to the vacuole, then slowly they get deposited and then it becomes like a hard cyst wall. So this was very interesting. So we wanted to see uh, okay, if we are detecting this marker, what about other genes which are expressed in Brady's whites? Because I showed you at the beginning that when tachyzoids convert to Brady's whites, they do some gene expression changes, right? So okay, before that we did the complementation. We can rescue the growth effect, and then we can also rescue we can see that the complemented strains do not deposit this marker on the walls. And when you look at the gene expression, we could see some difference between wild type and this mutant. So we looked at so we were expecting all Brady's white specific genes to be overexpressed. So when we looked at it, interestingly, so these are all existing data from literature, which is available in ToxoDB, by the way. And you can take, if you take Brady's whites, all these genes are overexpressed in Brady's whites only, but not in the other sexual stages. And if you look at this last column here is our mutant. Some of the genes are overexpressed, like here at the bottom you can see a few, but all the classical markers and many of the other important proteins like cyst matrix protein, they are not expressed. So this is like a in between sort of a phenotype. It's not a very clean Brady's white like phenotype, but because we got RNA data, we looked at all the other genes which are upregulated, and interestingly we saw all these genes which are usually upregulated in the sexual stages. These have nothing to do with Brady's whites. So all this family A proteins, CPW, WPC protein, domain containing proteins, and many of these other proteins, these are all sexual stage specific proteins. So that was interesting as well. So what's happening is when we made a RNA knockout in this stage, it's starting to express some of the genes which you will see only expressed over here. So we wanted to see, okay, what else is going on. So we just took all the genes that are upregulated in the mutant. So these are all the genes which are upregulated, around 600 odd genes. We looked at the profile of these genes in different stages from other data which is already available, and you can see that most of these genes are, the median value is upregulated. We took the downregulated genes from the mutant, and we could see that they're either not changing much or slightly, especially in the oocyst stage, they are coming down things. So it looks like we have stumbled upon like a set of genes which are controlled by this RNA. So this is like a summary. You can see here that if you take all these 615 genes and then compare it with these are upregulated genes. If you compare it with enteroepithelial stages and merozoites, these are the sexual stages of toxoplasma. You can see that there's a large number, 100 odd genes which are overlapping with genes which are expressed only in the enteroepithelial. And if you look at Brady's whites or sporozoites, they're not much. There are some overlap is there, but really not to this level. There is no overlap here. Okay, so that was interesting. And in the meantime, there is this literature which where people have worked on this protein called MARC. It's like a global regulator of HDAC3 activity. And if you see both this MARC and HDAC, they oscillate during the developmental cycle. So in tachyzoids and merozoites, they are high. Then when they go into the sexual stages, they slowly decrease. And then in the oocysts, they are less. And again, they come up because next stage is tachyzoids. 2 minutes. So in our system, we are seeing something like that. So we looked at carefully each and every stage expression. So this is comparison with the data which already exists, and you can see that if you look at the merozoite genes, again very few genes are upregulated in our mutant. Not the some genes are very highly upregulated, like you can see here, like this GRA81, which is very specific for merozoite, highly upregulated in this mutant parasites. And then if you look at the other enteroepithelial and gametes and all this, you can see a lot of these genes, whichever is upregulated in this enteroepithelial stage, in the mutant, it's all upregulated. And you can see all this flagella and all these dyneins, kinesins, which are usually in the gametes because they have motility and all these things. So these are now coming up in the absence of this RNA. And then finally, the oocysts which are shed by the cat, they have this lipids on the glycoprotein containing walls and all that. So we looked at the gene specific in these stages. So you can see in the oocyst stage, all these genes are highly expressed, and even in our knockout, all these things are highly expressed. And you can see some examples here. So, and on the contrary, if you take all the bradyzoid specific genes, you can see that classical marker like BAG1, which comes up very highly in bradyzoids, it's not at all there in the knockout, and most of the other classical markers are downregulated. So it looks like the story is okay. This is the last data thing, which is the transcription factors. All these transcription factors are important for sexual stage commitment, and they are all increased in moderately. Some are moderately increased, some are better increased in the mutant stage. So in summary, what I want to say is that it looks like the RNA what we knocked out, it is expressed in tachyzoids, and the RNA is important for tachyzoids to become bradyzoids. So in the absence of this RNA, they can't make good bradyzoids. But if you don't make bradyzoids, you can't get into sexual commitment. But in the absence of this RNA, the genes that will eventually be expressed in the sexual stages are already expressed in the tachyzoid stages. And so this is an interesting finding, and hopefully we'll solve the mechanism of how this whole process is working and what's the interaction between the RNA and its partner proteins or other nucleic acid. And maybe in the next meeting, I'll be able to say more. Thank you all, and I would like to acknowledge Sindhuri here, who did most all the work which I presented, and this is my group. Thank you all.
Thank you, Dr. Ganeshan, for an interesting talk. Now, this talk is open for 1 or 2 questions, please.
Danny, I was wondering, how would you distinguish between effects of the FAS1, which is the gene that your antisense is sort of embedded in, right? So when you knock out your non-coding RNA, how can you say that it's not because of the FAS1 that is causing these phenotypes?
Yeah, the FAS1 is anyway not expressed in tachyzoids. It's not an essential gene also. So we have done other knockouts in the locus. We can remove much of the FAS1 gene, and tachyzoids grow and bradyzoids. If you don't touch this RNA, if you do knockouts, I have that data. I didn't put it here, but if you just delete the first exon of the gene, nothing happens. The bradyzoids form, and you don't see this kind of a phenotype.
Great talk, Danny. So have you tried to overexpress this non-coding RNA to see what is the effect?
So I showed one slide where we did complementation. Okay. So we cloned this non-coding RNA with its native promoter, put it in one of the genomic locus, UPRT locus.
So what is the explanation why a non-coding RNA would affect so many genes? Is it possible that genome organization is affected because you deleted a chunk of the gene?
I had a very long conversation on this with Ali Hakimi. So
Dr.
current theory is that there are microdomains within the nucleus where there is organization of euchromatin, heterochromatin, and all this. So this RNA may be involved in organizing things. It may not be like acting like a transcriptional mechanism, but it may be organizational mechanism. So we are trying to, the next thing would be really to see where this RNA, is it like all over the nucleus, or is it present in some domains within nucleus and co-localized? Some things like that will help us.
So yeah, for getting into the
system, like it has to cross the strong intestinal barrier, like tight junction proteins, to get into the cells. So, what is the mechanism that anything that you know of that allows the entry?
So these parasites, they invade through the cell membrane. They don't go through the tight
it
junction.
goes through cell
The
membrane.
cell membrane. Actually, they have they bind through receptors on the host cells. The parasite has the ligands, and this receptor ligand mechanisms. Basically, it helps the parasite to attach to the cell, and then the parasite has the apical complex, which includes all these rophtres, micronemes, and then they secrete many effector proteins, which then make the barrier that breach the cell membrane, and they even create a vacuole through into which the parasite moves and it sits inside the cytoplasm.
Okay. I congratulate Dr. Ganeshan for your excellent talk.
Thank you.
Please come on this.
Thank you so much, sir. Now I would like to request our chairpersons to please felicitate our speaker as a token of appreciation.
The next speaker in the list is Dr. Hoti, former director VCRC Puducherry and current editor-in-chief for Journal of Parasitic Diseases, and he will be presenting on molecular xenomonitoring in lymphatic filariasis elimination programs. Dr. Hoti, please. Sir, the floor is yours. And you have 20 minutes.
Yeah.
Good morning, and at the outset, I would like to thank the organizers for this opportunity. You have heard enough about filariasis by Dr. Bhupendra Tripathi the other day. He has made my job easier. I am going to present our work on monitoring the elimination program using xenomonitoring techniques, which we have been developing for the last almost 2 decades. It's basically the story between the vector and the parasite. Filariasis, lymphatic filariasis, commonly called as elephantiasis, is a disease of poverty. 90% of the cases are caused by Wuchereria bancrofti, and the 10% of the cases are due to Brugia malayi and Brugia timori. And you can see the kind of morbidity caused by filariasis. You can see this lady has both the hands as well as legs are swollen, and it impacts the economy of the patient very drastically. You can see this particular lady, she is the bread earner for the family, but then her legs are swollen. In spite of that, she has to continue to earn the livelihood. So that's the kind of morbidity and economic impact the filariasis poses onto the affected individual. You can see these are the other pictures recently taken from the Jharkhand areas. So as I said, it causes stigma, it causes anxiety, and also psychological trauma. Then it affects the mobility, travel, education, employment, and marriage prospects of the affected boys and girls. So we can see here the globally the economic loss due to filariasis is about 2 billion per annum, and in India we have estimated it's about 1.5 billion. So the disease causes a lot of economic, social, and also stigmatic impact on the affected individual. Sorry. Yeah. It's a problem. Again, nahi move. Yeah. So basically, it is a childhood disease. The children get exposed to the infection during their early childhood, and by the time they realize that they have the parasite, it will be already too late. They would have developed the clinical symptoms. So in the sense, we have to catch them young to prevent the development of the clinical manifestations. As I said, there are 18 million infected individuals and 15 million have lymphedema or the elephantiasis, and another 25 million have the scrotal symptoms, which is hydrocele. So considering the morbidity and the economic impact caused by this disease, the WHO announced that this is the disease worth for elimination from the face of the earth. So in the year 2000, WHO launched the global elimination program using थोड़ा problem है. Next. वही. Okay. There is some problem, right? Yeah. So I think there is some problem. Anyway, there are about 357 districts in our country which are endemic for lymphatic filariasis. You can see all these red areas still have heavy burden of filariasis in the country, whereas some of the states have almost eliminated the disease. So as I said, WHO has launched elimination program, global elimination program with two arms. One is interrupting the transmission, that is to cut down the transmission of the filariasis in the communities, and the second arm is managing the morbidity of the existing cases. All the cases of lymphedema and hydrocele have been line listed in the country, and the interruption transmission is affected by the use of mass drug administration annually. There are two drugs. Initially, in the 2004, when India launched, it was only DEC. Then later in 2015, albendazole was added to the program, and more recently in 2022, another drug has been added to the mass administration, that is ivermectin. So now we have in some districts triple drug therapy, and that is going to be a major tool for elimination, but still we have a lot of challenges. Next. So this is the strategy. Initially, you will have to map all the endemic areas, and then the next step is introduce the mass drug administration in the community, endemic communities. All individuals have to be administered the drug, and then after 5 years of the mass drug administration, we have to go for the assessment of the impact, which we call it as transmission assessment surveys, and that is where we need appropriate tools. Diagnosis is one part, but monitoring the program is a major issue. Next, please. So there are 9 steps, like 7 steps in the marriage. So we have 9 steps in the elimination of the filariasis. These are the various steps and the tools required for monitoring at different stages. I have listed here. Next, please. In a sense, monitoring and surveillance is a key step in the control or elimination interventions. So, appropriate tools are required to monitor, and also then later stage certifying the program, whether the impact has led to the elimination of the disease or not. So that also needs a different kind of tool for making the decision for stopping. Okay. So the diagnostic needs for elimination program I have listed here. One is for individual case detection, and the second is monitoring the LFE program during implementation. What are all the tools required? Then for making the decision whether to continue the MDA or not, we need a different tool. And then post MDA surveillance, that is when you have very few parasite in the community, and even the antigens may not be there in the community. So we need very highly sensitive technique for monitoring and post MDA validation. So the tools that are available for filariasis as of now are microscopy, which has been used for the last 7 decades, and during 2000 we got another tool, antigen monitoring tools, that is immunological detection tool, and in late 1990s PCR, when that came into the arena, so that tool was also used for elimination program assessment. Next. Basically, when we talk of transmission assessment, we have to target 2 stages of the parasites which are depicted here. One is the infective stage, that tells us the actual transmission in the community, and the microfilarial stage, which is on the extreme right side, that tells us the presence of parasite in the community. So assessing the presence of these 2 stages in the community is very important for monitoring the elimination program. Next, please. So this is the microscopy. This is how you see the parasite. So this stage is the microfilarial stage, which you see in the human beings. So these are the diagnostics tools currently available. One is the OG4C3 ELISA, which is an antigen detection technique, and the other one is the RDT. So these 2 are currently available, but in the program only the RDT is used, whereas the ELISA is mainly restricted to laboratory investigations. But the problem what we are facing now is that even after 5 years of administration of the drug, still the antigen continues to remain in the population as detected by either of these 2 techniques because of the triple drug administration. So then how do we assess the program? If the antigen continues to remain in the community for a long time, how do you decide when to stop? So that dilemma is being faced by the program. Next, please. Next. So when we progress from implementation towards elimination, the sensitivity of the assay becomes more important, because in the community the parasite load becomes very low, even the antigen load also becomes very low. In that case, you have to have very highly sensitive tools for monitoring the program. So one way is you detect the antibodies which tell us the exposure of the community to the infection, or the other way is to detect the parasite in the vector. I will tell you why it is important to detect the parasite in the vector at a later stage of the elimination. Next, please. Next. So what we call detection of parasite in vector is called as xenomonitoring. So it is simply detection of parasite in an insect or the vector, that is called as xenomonitoring. And if you detect it by molecular methods, it becomes molecular xenomonitoring method. So this method has now been recognized by the WHO as an adjunct tool for monitoring the elimination program. Next, please. Basically, it was done by catching the mosquitoes from the field, bringing it to the laboratory, dissecting and detecting the different stages of the parasite. So by which you can arrive at infection rate and infectivity rate. Infectivity rate is very important for assessing the transmission, because that is the stage when the parasite enters the human beings. Next, please. So when we look at the dissected material, we look for the L3 stage, that is number 7, and the microfilarial stage, which tells us infection rate. Next. So here comes the importance of xenomonitoring. Dissection and microscopy, as pointed out by Dr. Anup a short while ago, that it depends on the seriousness, skill, and also peace of mind of the technician. If the person is agitated, we are likely to get less sensitive assay, and the lower layer, the infection rate will be very low. Not because of drug, but because of technician's seriousness. So that problem is there with the dissection and microscopy. And also in a program like LF elimination program, wherein 357 districts, the mosquitoes are collected from 357 districts, and they have to be dissected and examined. You can imagine the kind of work that is involved. So in that sense, it is very cumbersome and not suitable for, although it is currently being used because of there is no alternative. So we had to find some alternative method, and PCR came very handy in this regard. Next, please. So we developed at VCRC a very simple DNA extraction method from pool of mosquitoes. That is one advantage of PCR. You can subject the mosquitoes in pools, unlike which has been done in the case of COVID also. So you can subject the pools of 20 or even 50 mosquitoes per assay. So that is one great advantage of the PCR assays. So we have developed a very simple DNA extraction method, which costs almost few rupees. And we also have developed both for Wuchereria bancrofti and Brugia malayi. And subsequently, we also developed a RT-PCR assay for detecting the infective stage of the parasite in the vectors. They have very high sensitivity and also specificity. Next, please. So you can see here the cost comparison between the commercial assay and the assay developed at VCRC. The cost of extraction of one pool of mosquitoes is just about 25 rupees. Whereas the cost by using QIAGEN method is about 315 rupees. So that makes lot of sense when it comes to programmatic implementation of these xenomonitoring. Next, please. So this assay which we developed was validated in a multi-country fashion. About 8 countries were involved in this, and our assay was found to be very effective in detecting the infection in vectors. Next, please. Next. Here one important thing is that what kind of mosquitoes we should collect. We should collect only the gravid mosquitoes because they would have fed on the blood. If you collect naive mosquitoes which have not fed on the blood, you will not be able to detect the parasite. But then how do you how do you catch only the gravid mosquitoes? So we have developed a trap which gives us the mosquitoes which have fed on the blood, infected blood. Next, please. Give me 2 minutes, Chairman. Next, please. So this is the trap which collects the gravid mosquitoes. Next. Should give me extra because the snag in the text. Sorry. Next, we use an OB position attractant which is a dirty water, because this particular mosquito loves dirty water. So when it comes for laying eggs, so the mosquitoes are caught using the attractant. Next. So this is the kind of attraction we give to the mosquito for catching the gravid ones. Next. So this is how we set up the trap outside the houses for collecting the mosquitoes. Next. So the extraction method is very simple. You collect the mosquitoes from the field, bring them to the laboratory and dry them at 90 degrees centigrade or 100 degrees centigrade for about 3 hours. They become very fine, crispy like, and when we grind it, they become powder. Add tea buffer 100 microliter, homogenize, then boil for 10 minutes, centrifuge again, and your soup is ready for PCR. You don't have to use any clarification method or the high cost Cajun like kits for extracting the DNA. So the technique is very simple. As I said, it is validated in a multi-country fashion. I would request Dr. Anup to try this for Anopheles also for detecting the malarial parasites. Next. So this is the simple procedure we have for PCR. Next. So we did large surveys, and you can see the comparison between the conventional method and the PCR which we have developed. It worked very well in different case situations. Next. And we collected thousands of mosquitoes from 4 different districts and compared with conventional method of dissection and microscopy. The PCR method, the molecular xenomonitoring method which we have developed, is about 2-fold more sensitive than the conventional method. Next, please. We have also developed a PCR for detecting the infective stage. This summarizes our work on xenomonitoring. Now the WHO has identified it as an adjunct tool for assessing the impact of the elimination program. Next. So this is what I had to share with you. Thank you for this opportunity, and this is my very iconic slide of microfilaria from the blood.
Yes. Thank you, Professor Hoti. I think this was good review, and this was a good kind of alternative for eliminating filaria. I still remember during our PhD days when we were asked to make compounds for filaria, we simply shied away because the in vivo testing was almost 9 months it took to do it in Wucheria bancrofti and Brugia malai. So we always said no, we'll not do for filaria. But anyway, things are working for filaria now, and Brigank is doing wonderful job, and I think they are setting up the cell lines also to do the testing part. It's a good. So, if interested, only one question because of the time constraint.
Thank you very much for nice talk. And as one query, we're talking about the eliminations of this vector-borne disease, and WHO set up 2030 targets, many diseases, right? We have the problem related to the infection of co-infected in different diseases.
Co-infection with?
Co-infections like TB, HIV, and like the leishmania, many. But it's rare cases, right? So how we handle these cases for as a diagnosis or treatment?
No, you are talking about human infections or?
Yeah, human infections.
Right. Would you like to know about treatment or the monitoring?
Diagnosis.
Diagnosis, yes. Now, the recent concept that is being advocated is integrated diagnosis or integrated surveillance. So, wherein in an endemic area, if you have co-endemicity of more than 1 or 2 or 3 diseases, so we have to develop panel of a diagnostic which has a panel of different disease pathogens. So that activity is being now going on. So we have developed a multiplex assay for detecting the filarial parasite, malarial and visceral leishmaniasis, and also dengue and Zika. My young colleague has developed a multiplex to detect all these parasites in vectors, and that assay will be useful for human diagnosis as well. And my young colleague Dr. Saini is sitting there, who is also actively involved in this development of these multiplex assays.
Big cheer for Dr. Hoti.
Thank you so much, sir. Now I request the chairpersons to felicitate our speaker.
Associate Professor at IIT Mandi, who will present on the immunological roles of teniosolium, fatty acid binding proteins, and their potential as therapeutic targets in neurocysticercosis. And you have 20 minutes time with 5 minutes question times.
What is the okay. Good morning, chairpersons and everybody present in this auditorium. And thank you to the organizers for giving this opportunity to present some of our work which is going on at IIT Mandi. So, and excuse me for changing the title. Okay, so today mostly I am talking about how we can use these parasites. Like from last 2 days you have been hearing that parasites causing the disease, parasites causing so many economic burden, billions of daily lost, billions of rupees lost everywhere. But what these parasites are doing, only the losses. They don't have any other thing to do with us because they have evolved with the human for so long. Our immune system also recognize them. These parasites, and especially when we talk about the helminths, many of this they are called neglected. The reason is that these parasites are innocuous. They are present with us. They cause the morbidity, but very less mortality. So why they do exist, and how the human's immune system have incorporated changes among us or itself so that we can coexist. Is it possible to use this parasite for something else? Okay. The another thing which I usually take this opportunity when I go for a talk, that so when we talk about the neglected disease, some of the parasites or helminths are also called orphan disease. Very few labs you will hear that working on. Even if you say that from last 2 days, that in parasitic or in infectious disease, like called big 3,
Plasmodium, TB, HIV. Most of the fundings and research are directed towards that. But when we talk about filaria, schistosoma, tinea, very few research. But if you will go and see what is the disease burden, you will be surprised that close to 1 billion people out of 7.2 billion of the world population are in danger, and mostly like 25 million people are going through these parasitic helminthic infection. So this I also take opportunity to just give an awareness, and mostly like I know that most of you are a better scientists than me, so I will keeping the science part a little less and more about what are the challenges about these parasites, how which and what are the benefits. So starting with my acknowledgement, this is my team at IIT Mandi, and today whatever I will talk, most of the work has been done by these two good fellows, Suraj and Nana. And another thing, this is just an invitation that we are also doing a workshop for the systomics about the diagnosis and management of this disease, and you all are welcome to register, and registration is free, and this is very close by, 12 to 13 December. You all can come visit to the IIT Mandi. So coming back, okay, about this parasite. So if you will talk about the Tinea solium, okay, so we also call it the poke tapeworm. It is prevalent across the globe. Some countries are having more prevalence and called endemic, while others are less. Obviously, most of the developed countries, they have eliminated these parasites, okay. But at the same time, when you will see what is the incidence of the autoimmune disorders, so you will see that the countries who have eliminated them long time back, they have more autoimmune disorders, and those countries where the helminthic infections are still going on or the cycle is still there, they have less incidence. Maybe a coincidence, but nowadays a lot of people are working and have interest on the understanding the immunology of helminths, and it is coming up that okay, these parasites, what they do, the deworming, when deworming happens, they reduce the Tregs and increases the Th1. So whether that leads to the chronic inflammation, and that is the reason like okay, many of the autoimmune disorders are going high. So if we'll come to the India, like okay, there are few helminths which are very much prominently present, and among them, like mostly you will find that Ascaris and other diseases are there. Tinea also contribute to only 5%. And if we'll see the distribution of this Tinea solium infestation in India, okay, so you will see that few states are highly endemic, and many of the places you will see that okay, there is no report, actually nobody did any research, what is the infestation, what is the endemicity, and this is where these neglected diseases are. So the question is then, why is a parahelminth, which is only 5% infestation even in India, I am working on? So first reason is that this is our own problem. Very few places, very few countries you will find that people are working on this. Then another interesting thing, that now people are looking for a lot of strong immune modulator coming from the natural product. When we say that natural product, it can be plant, it can be anything. It infects muscle and central nervous system, and when you say that central nervous system, so this parasite when goes to the brain, it causes epilepsy, and you can understand when epilepsy is a big challenge. Understanding epilepsy is a big challenge, and when you talk about the challenge associated with the Tinea solium, so lots of you see that there is no, like, even as I have shown in previous slide, that many of the states, they don't have the data whether how much endemicity is there, what is the disease burden. Diagnosis, whatever the serodiagnostic tools are, those have been developed in Western countries, not working in most of the poor places like India, where co-infection, very high endemicity or morbidity is high. It is not going to work. And when you say that for diagnosis, what is the gold standard? The gold standard for diagnosis for central nervous system infection is MRI. And at the same time, we say that this parasite is endemic to the communities which are poor. So, and you know that the cost of the MRI, it's very high, so they can't afford, and there is no vaccine. Animal model is only pig, and you know that working with pig is not so easy. It's very cost and labor intensive. So challenges are very high. And even if you will say that what are the drugs available, so you must be hearing that even somebody which is working on filaria, they will say that okay, albendazole, for tinea, albendazole, and when it has been discovered 70 years back, and even if you will see that what are the mode of action of this drug, it's not clear. Now we have started this mass drug administration, and the another challenge is coming up, the resistance in the community about this drug, which is increasing, but not a single new drug is coming for the neglected disease in recent past, not in last 20-30 years. So we have lots of opportunity and challenges to work towards to eliminate, control in this way, or the further diagnosis of the tinea solium and neurocysticercosis. So a little bit of life cycle, because this is important to explain that this parasite lives in the gut, okay, and adult tapeworm comes through the human feces with proglottids. These mature proglottids are full of thousands of eggs. One proglottid is good enough for one community to infect because it will have thousands and thousands of eggs. These eggs can be taken up by the free roaming pigs. You know that in like poor countries where the micro agriculture or poultry is going on, they don't feed the pigs in their poultry farm. They keep them free roaming. When they consume, they get this cysticercosis to develop, and if it is eaten without proper cooking, it can go back and develop into another adult tapeworm. So this is called a normal cycle, teniasis. One dose of albendazole, you will be taking care of the teniasis. But another pathway, sometimes these eggs can go to the human through the water or through the raw vegetables, and when these eggs go directly go to the human system, sometimes again these eggs can hatch and pass through the intestinal lining of the gut and go to the blood. When it goes to the blood, this is small oncosphere, it can go any part, and mostly it goes to the muscle or to the central nervous system. Then it is called cysticercosis, and when it infects the central nervous system, then it causes the neurocysticercosis. Now the challenge is here that neurocysticercosis, when it goes to the brain, what is the outcome? The final outcome is epilepsy. And you know that how many taboos are associated with the epilepsy in Indian societies. Lots of challenges. And even many of the times, if you go to the communities which are poor, either they don't want to go to their doctor or clinician, or even they have wanted to go, they don't have the resources because it's costly. And when they go to the neurologist, he cannot assess or diagnose properly till the person is coming with MRI. And then again the cost of MRI, you all know, 8,000, 5,000. And many of the community family have this 5,000 monthly income. So the challenge is very high. So this is how the clinical spectrum of the parasite is that, like, even if many of the trained neurologists, it will be difficult for them to diagnose that it is neurocysticercosis without the proper diagnostic tool, because the clinical spectrum is very. It can have severe, simple headache to the multiple episode of epilepsy, and that's where the challenges are. And even in some extreme cases where the number of cysts are higher in the brain, mortality has been reported. And before the treatment of this disease, it is important that we should know the number and stage of the parasite. And then Why? Because this has different stages. If the parasite is viable or degenerating, you don't need to give viable, then only you need an albendazole to kill the parasite. If it is already degenerating, what happens at that time? Or you can see that they are causing edema. So already a lot of inflammation is going on. If you give more albendazole, there will be severe inflammatory reaction and the patient may have more episodes of epilepsy. So radio or diagnosis or MRI is important here. Even if the number of parasites are multiple and if they have been administered the albendazole, then again, like, because this can create a severe inflammation, so again it is said that don't give the albendazole. So for diagnosis and patient management, it is important to know the stage and the number of parasites. And right now, for these all things, the gold standard is MRI, and the challenge is that in rural setting you will not find this. So we are also working on that, how to develop a serodiagnostic tool which can say determine the stage and number of parasites. But today is like, okay, we'll be talking mostly about the immune, what happens, how it can be used for that treatment of other autoimmune diseases. So we have focused our study towards the ESP, excretory secretory proteome or secretome of the parasite. You know that like a lot of work is going on to understand the different kind of the proteins present in the secretome of different pathogens. So we also focused on the ESPs, and this is how the parasite, the cyst looks when you go for the, we isolate from the pore, just like white pearl, beautiful. And these are full of fluid, that is called cyst fluid, and they secrete a lot of you can culture, and they secrete a lot of proteins or the secretome. So we did that, we cultured the cyst and then extracted the secretome, and we started working with this. So these secretome was full of different kind of the proteins which have both pro-inflammatory, anti-inflammatory and some regulatory roles. So these secretes number of protein, and then we started looking what this secretome is doing. So one thing we did that, okay, this secretome can also be used for the diagnosis. Here you can see that, okay, they detect several specific band during the EITB, which is immune blot with the sera. So that can also be used for the diagnosis. And when we treated the macrophages with these ESPs, you can see that they give a mixed reaction, pro-inflammatory, anti-inflammatory, and this is very common with most of the parasites, especially helminths. And we look for what kind of the TLRs are responsible for recognizing different these helminthic ESPs, because there are previous report that TLR4 is associated with the occurrence of epilepsy during the neurocysticercosis. Here you can see that TLR4 all were significant except for TLR10. Okay, so then we started looking at the protein level, and you can see that the TLR response, the TLR4 was getting, when we treated the ESP, it was getting down. And when we looked for the different kind of the, because TLR4 is directly associated with the AKT pathway, we looked for the different activation of AKT, where you can see that when we stimulated the macrophages and human neutrophils both time, like, okay, what happened, that TLR4 was getting down, and the phagos we also characterize that this macrophages getting towards the M2 polarization. When TLR4 was getting, the phospho-AKT was getting down, we associated with the ROS activity, and you can see here that when the macrophages were treated with the ESPs, the ROS was getting down. And then we did all other kind, like, okay, what happened, where you know that when the M2 polarization is happening, the macrophages will be getting towards the less killing activity and less engulfment. And then we thought like, why this TLR4 is going down. Then we came to conclusion that epigenetic regulation. We came that, okay, some microRNA, especially MIR19, which is associated with the P10, have directly feedback mechanism, that is getting high, and that's why phospho-AKT was going down. And why less inflammatory reactions? Because this 145, which is 146 and 155, which is directly associated with the NF-kappa B activation, they were also getting down regulated, and that's why we came to conclusion that how this ESP is going to control the M2 polarization. So this is the final picture we came to, like, okay, when the ESP is there, they deregulate the phosphoic AT and they make the more M2 macrophages. Okay, so when this story done, that we said, like, what in the ESP? So we started looking, that in ESP we have the EVs, extracellular vesicles. So we started looking for what these EVs are doing in the vesicles. So again, what we did, we cultured the parasite and we isolated the EVs as per the standard MSI EV protocol, and most of the EVs were in the range of 100 nanometers. And then we found that when we did the GC, that many of the metabolites were present there which were associated with the AKT activity, and that gave us a good idea that what is going on. And when we look for further, like, okay, what phosphoic AT is going, we associated that this activation of phosphoic ATG is directly associated with the mTOR activity. We did all sort kind of, to establish this story, like, okay, from here onwards, like, if the AKT activity is going down, what the down regulation is happening, then we came like, okay, if AKT activity is going down, less ROS, less bacterial killing activity, more M2 polarization. So finally, if this M2 polarization is happening, so where we can use it? So we made a colitis model. And here, what I would like to just emphasize, that you can see this here. Control, you can see the pathogenicity, control the normal, and then you see that in colitis, how it is getting shrink. But when you treated the mice with the EVs, it's protecting. And same like we established with different kind of all the parameters which is needed to establish for this phenotype, that okay, when we are treating the mice with the EVs, you can see that with the severe pathology, when it is treated, you can see some of these are pre-EVs are almost close to the control. So what does it say? That okay, these parasites are present in our gut. They are taming the immune system, making them to survive, making them to cope up with the challenges. And these parasites proteins can also be used to control other autoimmune disorder. This is the story at present. We are also exploring the possibility of this EVs for the rheumatoid arthritis and cardiac disease also. So these all have been published. So forget about all the science. I know you all know better than me. The important thing which I go to anywhere say that please be careful while eating salad, half-cooked vegetables, especially you are endemic area. This is Lucknow. It's very endemic area. So when you are eating outside, anything like noodles, chow mein, momos, be careful. And this is our campus at IIT Mandi, and many of you will be aware that the next ISP is going to held in the Mandi. You all are welcome there to visit, to see our campus and collaborate also. Thank you.
Thank you, Dr. Amit, for the interesting talk. It was very, very excellent talk you have given. Now the talk is open for 1 or 2 questions, please.
Yeah, please.
Dr. Shamal Roy.
So nice talk. So you are basically driving at the hygiene hypothesis, right?
Yeah.
So